The last chapter that we are looking at in this semester is the extraction of energy from protein. And so what we're going to talk about is oxidation of amino acids. But in that, we get a whole different cycle and the production of one of the waste products that we produce, which is urea. And so there's an important part of amino acids that we need to take care of, and the urea is the outcome. Now, in terms of energetics, the carbohydrates that you eat and the proteins that you eat give about the same amount of energy per gram. Okay, so you can extract about the same amount of energy. Fats, on the other hand, are almost double, little over double the amount of energy you can extract per gram. But we have to break down protein in a very specific way. When you eat protein, you know that there's digestion that happens in the stomach. There are some proteases that are only active in the pH range that is found in the stomach. And so the majority of what happens in the stomach, though, for proteins is that they're denatured, opened up, so that they can be degraded. There are some proteins that aren't opened up, like the protein that is part of hair. If you eat hair by chance, it can get into your stomach and get into that very high pH, or actually very low pH, very high concentration of acid, and it doesn't denature, and it will not degrade, and it will not be digested, but most proteins will. The pH, that very high pH or very high proton concentration, doesn't actually break much of the protein down. It is the action of the proteases. Now, not all the proteases are active and not all the proteases are in the stomach. There are also proteases that occur in the small intestine in neutral pHs. And we are going to degrade the protein down into its individual units, which are the amino acids. Amino acids can be absorbed in the or through the intestinal wall. So individual amino acids are brought in. Now, we can break down proteins as part of our diet. We can also break down proteins as part of the normal process of biological systems. One, a protein is synthesized, it works, it gets old and ages, and it will be turned over. And every protein has a length of time that it's turned over. What we will use is a mechanism to tell how old the protein is, and then a mechanism to catalytically break it down. And so we can do that in an ATP-dependent process. Also, we sometimes will bring in proteins and degrade them in the lysosome and can degrade proteins. A lot of times the lysosome will be places where, like in terms of immunity, we will trap a protein that is foreign, bring it in, take it to the lysosome, break it down to disable its function. But then those proteins that were coming in to infect us can actually then be broken down into amino acids and then broken down into energy. So the lysosome and the proteasome are places where we have additional breakdown. Now, turnover, where a protein is synthesized and then it lives for a little while and then it becomes too old and it gets recycled. Well, how is the aging process done? It's done by having a small protein added to it called ubiquitin. It's a 76 amino acid protein, and it gets added over time. Ubiquitin gets added to all proteins, and one ubiquitin, you're okay. Two ubiquitins, you're still okay. But over time, ubiquitin adds, ubiquitin adds, ubiquitin adds. Long enough, it will then be tagged to be taken to the proteasome. Usually, it's degraded in the proteasome into peptides between 6 and 10 to be degraded further into individual amino acids. It takes up to 4 or more ubiquitins to be added before it's tagged to go into the proteasome. Now, once we have single amino acids, there is something unusual about that. If we have extra sugar, how do we form a storage molecule of sugar? What is the storage molecule of sugar in the body? Glycogen. If by chance we have extra fat, where is that stored? Adipose tissue. If you have extra protein, you cannot randomly just put them all together, because when you put proteins, the amino acids together, you create a protein that could potentially have function. There is no storage molecule for excess amino acids. Now, amino acids could be charged. Another way to say that could be added to transfer RNAs and could be a source for new synthesis later. But what happens if you've eaten so much protein? All of the transfer RNAs have an amino acid associated to it. You are not signaling protein synthesis, and you still have extra amino acids. You must degrade them. Okay? This is part of the idea of the Atkins diet, or high protein diets, is that we will start the process to break down the protein, and when you break down protein, quite often you will also mobilize fat breakdown at the same time. All right. Now, for it to be transformed into usable entities, the parts of it, there are really two parts of an amino acid. One, the amino, and the other, the acid, yes, the carbon base. Okay. The ammonia that is on the amino acid, though, is and can be toxic. Okay? If you just cleave off ammonia, it becomes the ammonium ion because of pKase. And if it is in the ammonium ion, that's something we see all the time, ammonium. If it gets into a very low concentration, it actually becomes poisonous to us. So we can't just take off the amine just randomly. We have to take it off in a very controlled manner, not allow it to just float away and build up in concentration, but we have to control where it goes. And so the amino acid, you will have the amine, you have the alpha carbon that has an R group on it, and this would be a time to go back at look at your 20 amino acids and know what those 20 R groups are, and then the acid group itself. When we have turnover, we're going to break off the amine, okay, heading to the urea cycle. And then we're going to break off the carbon skeleton in what's called an alpha keto acid. An alpha keto acid is an acid that is alpha to a ketone. Okay? We have seen a couple of molecules that are alpha keto acids already. One of which is, if you put a methyl group here, pyruvate is an alpha keto acid. Alpha ketoglutarate is also an alpha keto acid. Both of them were synthesized by a dehydrogenase complex that was very complex. And so we've seen a couple alpha keto acids that roam around. But when you deaminate any of the amino acids, take off the amine, we do it in a way that we replace the amine with a ketone, and it becomes an alpha keto acid. Once we have the alpha keto acid, it will break down into one of the pathways, into one of the structures of one of the pathways that we have already learned, most of which will be the citric acid cycle. But we will also go into other pathways. All right? So, cells convert, you digest all of your amino acids. Amino acids transported to the liver. They are interconverted. And what we do find is that almost all amino acids, before they actually get broken down, get converted into one single amino acid, glutamate. before it gets broken down. All right. And we get energy from muscle breakdown. And that's where we can get energy from. So we can eat it, we can convert it from others, and then we can have muscle breakdown. All right. Here is where the big thing happens in the first step, where we transfer everything and end up making glutamate first and then into glutamine. When we start the degradative process, we start off with an amino acid. We will then use an enzyme called a transaminase. You'll see that in just a little bit. What the transaminase does, it uses, it uses an alpha keto acid, most of the time alpha ketoglutarate. It will react with the amino acid. What it does is it exchanges this, that ketone and this amine become this amine and that ketone. They change places. Okay, so this is an alpha keto acid that comes in, a different alpha keto acid falls off. Okay, just has a different R group. The amine comes in, and I have amine stuck here, and now I have glutamate. Okay, so to get rid of the nitrogen, most of the time the nitrogens are given to glutamate originally. Now, glutamate can react one more time and accept one more amine in the R group to become glutamine, and that's kind of a buffering system if I have lots of amines coming off at any one time. Most of the time, it is through the process of glutamate that we create the free ammonium ion, and that ammonium ion goes into the urea cycle to make urea. There are a few amino acids that can release ammonium directly, but there is a transaminase for every amino acid, and some of them can deaminate by other mechanisms. One that gets moved around quite often when you have protein that is being mobilized, and we look for alanine. Alanine is a protein that gets mobilized quite frequently, and alanine can be converted into a intermediate that can be broken down into CO2 and water. But alanine has a special ability to have its carbons made into pyruvate, and then from pyruvate through gluconeogenesis, it can go to glucose. So protein carbons have the ability to make glucose. This is important because when we're under starvation conditions, we want to make sure we have enough glucose to keep the brain functioning. We cannot utilize any fat energy to do that, but we can use our protein to do that. All right. In the degradation of those amino acids, the nitrogen is converted. And so the general enzyme is called aminotransferase, and then you have aspartate aminotransferase, alanine aminotransferase, and all of the amino acid transferases. And you can see a substitution of the nitrogen for the ketone. All right. Now, we find aminotransferases, there are 20 of them that exist in nature. Most animals have only 18. A couple of the amino acids can deaminate in a much easier way. But we do find, and we do have 20 full transaminases that are in nature. The aminotransferase is going to use the cofactor PLP. We have seen PLP before. We've seen PLP used as a cofactor for phosphorylase, where we broke down glycogen, bringing in a phosphate, and the phosphate breaks the glycogen, and glucose 1-phosphate falls off. The whole reactive species for the PLP used in that for glycogen phosphorylase was this phosphate to orient the incoming phosphate that's going to react. And transamination doesn't do anything, okay? In transamination, we're going to use the ring system, okay? We're going to use this part of the reactant to help facilitate a breaking of a bond, all right? The vitamin B6 is the precursor to PLP, and we have the functionalized PLP as the active cofactor. This is a long mechanism, okay? PLP is linked to the enzyme originally through a Schiff's base. What is a Schiff's base? Carbon double bonded to a nitrogen. Usually you have a positive charge, okay? And so what really happens is a ketone or an aldehyde reacts with a free amine to form this. If I take a look, here's where that cofactor is going to react with a lysine inside the enzyme so that it is covalently linked into the enzyme itself. So originally, the cofactor is covalently linked. There are other amino acids around in the active site to help facilitate the reaction. And so this is showing an arginine in that particular position. All right, here we go. Originally, the enzyme has a particular lysine. Here's the enzyme, here's the lysine. I have a nitrogen that has a positive charge double bonded to the carbon. This used to be just a carbonyl. Then I have the amino acid come in. All right, well, how in the world do I get these two things to look like this? All right, well, first off, looks like I need to articulate all of the hydrogens right here, because in the product or the intermediate state, I have 2 protons, where in the reactant state I have 3. And so I will first deprotonate one of these protons, and I will take the pair of electrons and attack the nitrogen. Okay, and something is missing. There is something missing. Okay, that has to be there, because when I attack that, this has to pick up a proton. All right, there's the intermediate state. The next reaction that occurs, the intermediate has 2 protons. Here it has only 1 left. Okay, and at that point in time, I'm going to have the enzyme as the leaving group. All right, what did that whole series of reactions do? The cofactor was once connected to an amine, that was the protein itself. Now the reactant's amine is in the same conformation, the same bonding as it once was with the protein. From the PLP's perspective, it really didn't change much. It's still attached to an amino acid, right? Still attached to a Schiff's base, but now this makes the amino acid much more reactive than it once was. Okay, the last time the lysine reacted with its R group and it had just methylenes, but here I have a different set of reactions. I have a carbon and nitrogen, that's the same, okay? I have a carbon and nitrogen, that's the same. Then I have a carbon, but this whole functional group is different. All right, this is set up to allow us to break a bond. Okay, ultimately the bond we need to break is right here. Sorry, it's right here. We need to get the amine away from the incoming amino acid. All right. This is what facilitates the reaction, because I can now invoke that ring system. Deprotonate. This pair of electrons forms a double bond. This kicks this double bond down. Then this kicks this double bond down, and this kicks that pair of electrons onto the nitrogen, making it neutral. I now have a lone pair sitting there. All right. That's actually a pretty stable intermediate, because beforehand I had 2 positively charged nitrogens. Now I have 1 positively charged nitrogen. Not too bad. Okay. Now the reaction reverses in the electron flow. I come back, I reform a aromatic system, but instead of going all the way back, this pair of electrons, instead of going back to where it began, will now pick up a proton. So the ring structure now has been used, and I have a Schiff's base between the nitrogen and the carbon of the amino acid, where the Schiff's base was somewhere before. Now, Schiff's base can be broken very easily by adding water. That's what we're going to do. Okay, so my next reactant, I am going to have water as part of the reaction. And then I have a base take away a proton. I will react at the Schiff's base, and I will pick up an additional proton. So the nitrogen now has 2 protons. On top, there is a hydroxide attached. And instead of having the nitrogen bound to two bonds to the carbon of the incoming amino acid, now I have 1. In one last step, I will deprotonate, form a ketone, which used to be a carbon bound to a nitrogen. Now it's a carbon bound to a ketone. And this, oh, I'm not going to pick up. I'm going to show it as, I'm going to just drop the electrons onto the nitrogen. I brought in an amino acid. What can float out is an alpha keto acid. The specific one depends upon what amino acid you brought in. Big deal. Right here, the amine has been taken off the incoming amino acid. Now, what I said in transamination is we have an amino acid, we take the amine off, but we give it to alpha-ketoglutarate to make glutamate. We've done half of the reaction. We took the amine off, but now that amine needs to react with alpha-ketoglutarate. Now, this is an alpha-keto acid that can float away. And what can float in is an alpha-keto acid, but the specific one is alpha-ketoglutarate. Okay? From this point on, the reaction is exactly the reverse steps, and what will be the thing that leaves in the reverse is glutamate. Okay? And overall, I got you to right here in my steps of drawing. The the alpha-keto acid that came off the amino acid. And this one is the alpha-ketoglutarate. Alpha-ketoglutarate. It will do the reverse steps, the reverse steps, the reverse steps, and the specific amino acid that comes off here is glutamate in the stage 2. Okay, that is a whole lot of work to move this around. All right. Aminotransferases are important in breaking down protein, but it's also important when you do blood panels. Okay? If you do a blood panel, one of the things they actually look for are aspartate and alanine aminotransferases. If by chance you see the proteins that break down amino acids in the bloodstream, okay, that indicates you either have heart damage or liver damage. Your cells are breaking down, opening up, and putting these into the bloodstream. You should have very little of these just in normal healthy blood. And so a lot of times if you're looking at liver damage or you're indicating some liver damage, these are high in the bloodstream, or these are something that you look for if you have a stroke or a heart attack. And they can be monitors of damage to cells or cell death. Okay. Why in the world do I give the ammonium just to glutamate? It is for the use of this one particular amino acid, this one particular enzyme, glutamate dehydrogenase. Glutamate dehydrogenase is as you can see by the name, an enzyme that will have oxidative reductive types of reactions. And so what we do is we've got 3 protons, and in the end we've got 2. We had 1 here, and we don't have any there. And then accepted by NAD plus, and we get, oh my goodness, another Schiff's base. And we know if we add water across the Schiff's base, what you can think of is this. If I add water, okay, the bottom becomes a ketone, and the top becomes NH3, and if it's just a hydrogen, it'll be NH4, but that comes off. So when you break a Schiff's base with water, you get a ketone. That doesn't look like a ketone. Let's erase that. You get a ketone at the bottom, and then you have a free amine falling off. This is the one in which we release the poisonous ammonium ion. And so we control when this one actually reacts. Okay, this enzyme actually has the ability to use NAD plus or NADPH. It is so fundamental a reaction. That is unusual. Usually the NAD plus is the acceptor. Usually NADPH is a donator, but this one can actually occur. This reaction is regulated by energy charge, or how much energy you have as measured by, do I have a lot of ATP around, or do I have a lot of ADP? Or we also use GTP as an indicator. The 2 amino acids that can actually deaminate by themselves without an aminotransferase is serine and threonine, because if you can dehydrate them, take a water out, a proton and a hydroxide, you create the Schiff's base. And then you add water across that, and the amine can come off. Both of them, both the hydroxide containing amino acids that are aliphatic, can do that. All right, once the ammonium is created, we need to very quickly segregate it and get rid of it. And so what we're going to do is we're going to send the ammonium into the urea cycle. One interesting thing about the urea cycle, it is the first metabolic pathway in the body to actually be described in full. Okay, it's kind of odd and a whole lot more work when it is also a cyclic intermediate. So somebody chose the worst one to start with, a cyclic one, but this is the one. Okay, the urea cycle, one of the nitrogens of urea is derived from the ammonium that is released from any of the amino acids, and one of the other ones comes from aspartate. But you got to remember what urea looks like. Okay, urea looks like this. What color is urea? It is clear. You may think it's yellow until you actually go into lab and actually work with it. Has anybody ever dissolved urea so that you can denature proteins? What is it when you dissolve it? Did you actually feel the test tube while you were dissolving it? No, this one actually really gets cold. A lot of times cold packets that you have, the emergency cold packets, have urea in them, and it's a very endothermic process for it to dissolve. To denature proteins, how concentrated did you make your urea?
I think it's 12.
Yeah, 12 molar. Yeah, and that's pretty hard to get to too. Very high concentrations of urea. All right. And so we're going to take urea into the cycle, and man, what does this show me? I'm looking at this slide here, and it's complicated. And it doesn't really tell me much. Okay, all right, what in the world? We have just looked at this. Take an amino acid, bring in alpha-ketoglutarate, it gets to glutamate, and we produce the NH4. That's what we've learned up to this part. So the last thing we did was we made NH4. Okay? Before it can get to the urea cycle, the ammonium must react with bicarbonate, which is a dissolved and reacted with water, CO2. But check this out. That's costly. 2 ATPs are needed to get this into the activated form called carbamylyl phosphate. Let's take a look at that reaction in a little bit more detail, and just a little bit. Here is the cycle itself. Okay? We make the carbamylyl phosphate where this is the nitrogen that had just come off. It's going to react with a molecule called ornithine to make citrulline. This reaction occurs in the mitochondria. Citrulline is transported out into the cytosol. It will react with aspartate to create arginiosuccinate. Arginiosuccinate will react, give off fumarate, and produce arginine. Have you seen fumarate before? What cycle can this molecule be entered into? Is this a citric acid cycle as it's being thrown off right here? Is this in the citric acid cycle? Why is it not in the citric acid cycle? It's in the cytosol. Citric acid cycle happens in the mitochondria. So that has to be transferred. Arginine then gets reacted to form urea, and I reform ornithine. So it is cyclic in nature. Before we can actually get into the cycle, we take bicarbonate and react it with ATP. What is wrong with the mechanism as drawn? Kick up, kick down on the double bond. All right. The ammonium comes in and can attack. The phosphate is a good leaving group. The next reaction is again attacking ATP at the alpha, beta, or the gamma phosphate. How would you know if it's the alpha, the beta, or the gamma? What's the leaving group? ADP. So I only transferred one. It had to have been at the gamma. And I get to my carbamylyl phosphate. All right. All right, the carbamylyl phosphate reacts with ornithine. Okay, first off, classify what kind of molecule this is. If you had to put it into a big category, what category would you put that one in? It's a what? It is an amino acid. Is that one that you learned? No. It is not one of the 20 amino acids that are attached to transfer RNAs. But an amino acid is a much more general molecule. It has to have an alpha carbon, a hydrogen, an acid, and an amine, and then some sort of R group. So this is an amino acid. What does it most closely look like? Which amino acid? It looks like lysine. It's missing one carbon. Okay? And we're going to do a reaction. All right. You always have a base and an acid to use. All right. We are starting with the base because it's all about the no trouble. Okay, great. Then what happens? Okay, good. Oh, that's interesting.
Yeah, that's kind of where we got stuck.
Okay.
Because we only have one over here.
Yes.
Starting with 3.
Correct.
And then this is an inorganic phosphate.
Well, use a little pointer so people know what you're pointing at. The little dot. Yeah, that doesn't. Okay, so what was your issue?
So we have
Only one there.
hydrogen over here. We have 3 right here.
Okay.
And so 2 have to leave.
Yes.
And this is an inorganic phosphate that's leaving, so it has a hydrogen on there.
Okay. Yes.
And so we don't know how to.
So that last arrow point with that last arrow movement, you wanted to go ahead and take care of that proton and have the inorganic phosphate fall off. That is not an unreasonable thing to draw, although that phosphate will not be the one that takes off. Not only are we in an aqueous system, we're also in a buffering system. So once the phosphate leaves, if you didn't do that last attack to take off that last, you would actually have an extra proton there. But the pKa would be so high versus the solution that the buffering system would take it off. The inorganic phosphate falling off would have a pKa so low based upon the solution that it would pick up the proton from the buffering system.
So it's like that.
So if you go up, go down, you could
And then you have to
kick it on to, if you kick it on, if you actually
I'm scared of drawing it again. Someone draw the last one. Okay.
draw the little last arrow. The little last arrow. Okay, there you go. Okay, if you did that, you would need to draw an intermediate and show the other, or I would be okay to interpret what it is, but for you to understand, you might need to draw the intermediate and have a base take off the proton, and then you would have an acid to be picked up by the phosphate. The first drawing would be acceptable because I know what's happening. I don't want it, though, that first drawing to confuse you because it really isn't the phosphate taking it off. But those deprotonations and protonations actually have to happen. So that was a good mechanism. Thank you. All right, who was number 2? Okay, number 2, and who is the people that can help number 2? Okay, this is your help group that can. Okay, so there you're going to articulate the.
We have some electrons on it.
I've got electrons on that nitrogen. Okay.
We have to somehow. Our problem has been
Uh-huh.
where does it what does it attack? We know it attacks at the alpha phosphate on the ATP,
Uh-huh.
but we don't know what attacks.
Don't know what attacks. Okay.
So
Oh, wait a minute. Interesting. Okay. That.
that'll go attack.
Oh, wait a minute. There might be some. There may be an intermediate that's weird.
Because initially we were thinking of base, but we couldn't figure out how, like where that extra hydrogen.
Where the extra hydrogen came. Wait, yeah.
Yeah, because otherwise we would have said that it would have attacked the hydrogen on that.
Oh, okay. Wait. Yeah. Okay. Yeah. Okay. I got it. Okay, you don't. Okay, all right, watch this.
Okay, go for it.
Okay, check this out. Now, helper group, do you got a help for them? Okay, what's your help for them?
The lone pair is going to form a double bond that's going to kick up, and then those electrons on the oxygen are going to attack the alpha.
There, and then that.
Oh.
Oh.
Okay. All right.
You actually had a real good helpers.
Yeah, we did.
Woo! Pulled that out. And now the oxygen is negative, and that can be the nucleophile. Now, where is it going to attack? Okay, be careful.
Pick up, pick down, and then I guess this one.
Yes, perfect. All right, now you got to the intermediate. Good, you're halfway there. Do you need more help? Are you okay now?
There's no help to give.
There's no help to give. Oh, come on now.
We were mostly focused on this first part.
Yeah,
I worked on this last part.
and this last part now is, why do you add an AMP? Why do you add an AMP to something? Good leaving group. So you attack something that usually is a carbonyl. We don't have a carbonyl here, right? But we have something that looks like it and acts like it. So what has to attack? So there is a carbon over there that has 2 nitrogens attached to it, right?
Yeah.
Okay, use the highlighter and show me where that is.
So,
It also has an AMP on it, right?
yeah, so this is part.
That right there, okay? Look in the product. Where is that carbon?
That carbon will be right here.
Yes. Okay, so what has to attach to it? One more what?
One more nitrogen
And where do you get that from?
from right here.
Yes. Okay.
So there's, okay.
More gooder?
So you're going to have a base. It's going to deprotonate this.
Yep.
Right? And this is going to attack up here.
Yes. Now I hit a double bond, so if you have a pi bond, it has to.
Pick up, pick down. That's awful. This has to go pick up a hydrogen.
Yes, that is a good. And you have almost got to your product. What is additional thing that has to happen? How many hydrogens on the nitrogen that came in and attacked at the product? Only 1, but you still left 2. It's a pKa argument. It would be so acidic that, so basic that the it'll be deprotonated. So, but that is the mechanism. Very good.
So, when it kicks up and kicks back down,
Yep.
is that breaking the bond?
Breaking the bond between the carbon and the AMP.
Okay, so what electrons are grabbing that random hydrogen?
The bond, between the carbon and the O, that pair of electrons is grabbing that proton. She's the wind beneath your wings. Very, very good. That's the key.
Yeah, it's think about that.
Beautiful. Perfect. No comment necessary. Drop the mic. All right. No, not drop the class. Drop the mic. Oh, sorry. Say that again. Okay, so they drew that the base took the The base took the hydrogen formed the double bond, which is this double bond. This bond broke. They wrote, put it on the nitrogen, then pick up a proton. I'm lazy. I would have probably drawn that. Both of those work.
Organic makes more sense now.
That makes me feel so good.
Yeah, I feel like
There's a reason for why organic happens now. It's not just a bunch of memorization. There should be a reason why things happen. There you go. We now have urea and ornithine. That is perfectly correct. All right. Woo! We got through that. Do you feel it? Sure. Okay, as you approach these problems, okay, what strategy do you use to approach it? I know you first look at it and cry. Okay, after you do that, you draw a base and you draw a hydrogen, right? Proton. That's good. Figure out what's different. Inventory. What between what is changed. So it identifies one little tiny place in the very large molecule where the reaction is actually happening. Everything else is just R. Okay? Don't let the big molecule confuse you. Just look for the things that are changing. Sometimes you see it really, really easily, and sometimes it doesn't come as easily. And hopefully you're starting to see some principles about why something might be acidic, or if I've placed a phosphate or an AMP, that's going to be a leaving group in the next step. And the carbonyls, or a double bonded to a carbon, that's usually a place where the nucleophiles attack. And so we're starting to use principles. Where do I attack the ATP? Is it on the gamma phosphate to transfer 1 with ADP as a leaving group, or did PPI come off and AMP transferred? That was an alpha attack. So you're employing a lot of strategies that we've seen. All right, the overall reaction looks so much simpler. CO2 plus an ammonium plus 3 ATPs plus a aspartate plus 2 waters makes urea, 2 ADPs, 2 PIs, 2 AMPs, 1 PPI, and 1 fumarate. You can also see it this way in some books, because PPI gets broken down immediately by inorganic pyrophosphatase.
Did you say 2 AMP?
No, I think it's 1 AMP and 2 ADPs. Okay. All right. Now, there's what's called the urea and citric acid cycle bicycle. Ooh, that was creative. Okay. Well, what in the world is the bicycle? Well, one of the reactants is aspartate. Okay. And one of the products is fumarate. Well, fumarate can be converted into malate because malate can be shuttled inside the mitochondria. Once malate is inside the mitochondria, we have the citric acid cycle, a cycle that I know. And now you know urea cycle. The urea cycle, malate reacts to form oxaloacetate. Oxaloacetate, if it undergoes transamination, becomes the aspartate. And that's the aspartate that reacts. And so the aspartate, the fumarate, the malate, the oxaloacetate creates a cycle that connects the citric acid cycle and the urea cycle together. Okay? And so oxaloacetate now becomes a very important molecule. It can react in the citric acid cycle. We know that oxaloacetate is also a pathway to start gluconeogenesis. We now understand that oxaloacetate can also react to form spartate, which is part of the urea cycle. And so this particular intermediate, the oxaloacetate, which is an alpha keto acid itself, is very important. Defects in the urea cycle. Not something you really want to have happen, but there can be a couple ways in which a deficiency in one of the enzymes, we can actually treat it. Okay? If not, I said a very small amount, 60 micromolar is the amount by which ammonium starts to become toxic in the blood. So that's a very small concentration. Okay? There is the arginiosuccinase deficiency. If I can't break down arginiosuccinate to make arginine, which can't make urea, okay, that seems really, really bad. Now, what in the world do? We treat by adding large amounts of arginine, and we limit the intake of protein. Why in the world do we do that? Okay. Well, this urea that is given off in this person has nothing to do with the amines that are being broken down by the person. Okay, the amino acids themselves. The urea is coming from the arginine. What we do is we give a ton of this so that it will create ornithine, and so that it will take the ammonium that is coming off the degradation of protein to create citrulline, and we build up arginiosuccinate, which becomes the waste product. We create a linear path, which this is the waste product, rather than a cycle anymore. Okay? Now, if I make a linear path, I could have treated with arginine, or I could have treated with ornithine, because I just need a linear path. Why do I use this one rather than ornithine? Breaking down amino acids is kind of dangerous in a person like this, because we don't want to make an intermediate that is not normally taken out of the body. Why would we use arginine rather than ornithine? Mechanistically, are they very, very different? No, not a lot. So it isn't a chemistry issue. What other issue could it be if it's not chemistry related?
Well, it's used in proteins, so if you get too much of it, can you use it to make proteins?
If you get too much of which?
The arginine.
The arginine? Well, as long as arginine is in there, it can be processed into urea, and that's normal. And so that's okay. Arginine can be reacted that way. Arginine can also be reacted with transaminase, and you might build up a little bit of this, which might be a little bit bad, but it's more simple than what you think. Yes, it's access. Where do you find ornithine in large quantities? Not a lot of places. It's an intermediate in the cycle. Where do you find arginine? A whole bunch of places, wherever protein is. And I can isolate arginine very easily. It's cheap. It's kind of the answer of why we do certain reactions and processes at Lee, because we have cheap reagents. What can you do with a cheap reagent? Okay? And so it's a cost issue. All right, there are a couple other places that we can have deficiencies. We can either have the deficiency in incorporating the ammonium into the first intermediate, or we have a deficiency in the enzyme that takes ornithine and makes it the citrulline so it can go down the cycle. The treatment for these is to take benzoate or phenylacetate. Benzoate can actually react with glycine itself, and the phenylacetate can react with glutamine itself, both of which create molecules that can be excreted. Okay? And so, making sure we get rid of the urea is very, very important. From this point on, we've invested energy to break down amino acids. Now we can start to extract energy from the amino acids. Now, I said carbohydrates and amino acids give about the same energy. Well, we didn't have to invest a lot to break down carbohydrates. Okay? But here we have to invest some energy to get rid of the ammonium. The carbohydrates, when I bring in one, I got 6 carbons. Okay, and I can extract an amount of energy. With the amino acids, I have some that have less than 6 carbons, but I have some that have more than 6 carbons. And on the average, I will get more carbons than 6 per gram than I would have from the carbohydrates coming in. Also, most of the amino acids are more reduced than the carbohydrates themselves, so there's more opportunity to oxidize them to extract hydrogens. And so I can, even though I'm taking a lot of energy to extract them, to take that amine off and deal with it, I do net more energy later. Okay. Have a wonderful afternoon.